Good evening, Ashrita. How are you?
Good evening, ma'am. I'm fine. How are you?
I am also good. Thank you. Let us wait for the others also to join.
Yes, ma'am.
Last time we have discussed about the discovery of cell, right?
Yes, ma'am.
Okay. Why they have not joined? ये बात नहीं भाई। भैया मामा ऐसे आने काम करने Okay, I think we can start. Medha and Ashrita, turn on your video. Hi, Medha. How are you?
I'm fine, ma'am. And you?
I'm also good. So, Medha, you are sending each and every homework, which is very, very good. Ashrita, what about you? Your homework?
Thank you. Ma'am, I have also been sending homeworks.
Yesterday's, have you sent it?
Yesterday,
Though, okay, that you have sent only, I think you have not solved it. You have directly sent the answers.
yes, ma'am.
You have to solve and send it, okay?
Okay.
Fine. So, because then only sir can guide you, right? Where what step is right, what step is wrong.
Okay, ma'am.
Yes, Geeta is also, Ashita is also sending. Okay, good. Fine. Good evening, Geeta. Today you are late.
I directly came from my class. It became a bit late, ma'am.
Oh, okay. Okay, fine. Just a minute. Okay, so last time we have discussed about the discovery of the cell. Now we will be discussing the next point, which is about, uh,
Um,
yeah,
I
tell
have
me.
uh, on Sunday we only have a science test.
Yes, one test in a week.
Okay.
Okay, so science will be there. Okay, now what should I discuss? Last time we discussed about the discovery of the cell. Robert Hooke, he was a person who discovered the cell. And today we will be discussing about his microscope, which was not very developed, but yes, he tried to use his compound microscope to give the identification or to discover the cell. Correct? So let me share the screen so that I can take you to his microscope, what kind of the microscope it was being used. Though it is not given in your book, children, but yes, I hope like in the future you're going to write the entrance exams, right? So for those part, it is important to understand about the working of the microscope, which was used by him. Okay. So two microscope we will be discussing. One is which was used by him in the year 1665, and the other one, which is like we are using in our school right now currently, which is called as the compound microscope. Okay. So let me share the screen and do let me know, children, whether it is visible or not to you. Is it visible?
Yes,
Yes.
ma'am.
Okay. Huh. Fine. So I'll just duplicate this. See, this is the microscope. Can you see the microscope? What was used by him? Is it
Yes.
visible, children? Geeta? Okay, fine. Now here, this is the okay. I'll tell you first of all basic idea. I'll give you. So during that time, as I told you, the microscopes were not invented, were not discovered actually properly. So he just used his simple microscope to work on the cell. So this is the microscope what is given in the picture. And it was a kind of the compound microscope only where you used two lenses for the magnification. Now, the top part of it is called as the eyepiece. Eyepiece, why do we call it as eyepiece? Because through the eye, at this area only, we will be keeping our eyes and watching the specimen. Have you seen the microscope, children, at your school, or have you used it at your school? Ashrita, Meda, and Gita? You have, right? So
Yes,
what
ma'am.
you will be doing, you will be closing actually one of your eye, and through the second eye, you will be looking into it. So that is why it is called as the eye piece. So to look into the object, to look into the specimen, we will be using this eyepiece part. The second part here, it is called as tube. It is like, the center part of the object. So this is a microscope. So the center part of that microscope is called as a tube, which will be connecting the lower part with the upper part of it. This one. The objective lens. So basically, objective lens will help in magnifying the object. Do you know, children, what do you like? What is magnifying? Any idea about magnifying?
Yes.
Increase the power.
Okay, power will be increasing. Okay. Now, if I say that we have 10 magnifying power, we have 20 magnifying power, which is more power according to you? Which is more powerful, 10 one or 21?
20.
Very good, Medha. Very good, Ashita and Geeta. Correct. 21 is more powerful, which is correct. But now what happened? A very important point I'll tell you. They are the different kind of the lenses. There are the different kind of magnifying power. Okay, which we will be using it here. Generally, we'll have the 10x, we'll have the 15x also, we will have the 45x also, we will have the 90x also. So in our microscope, we will be getting such kind of the magnifying power. Here, the capital X means magnifying power. Okay, like how you're writing centimeter, meter, millimeter, all those things. The X, capital X is written as the magnifying power here. Now, 10x, 15x, 45x, 90x. Definitely 90x means highest magnifying power. But directly we cannot use the 90x. Why? I'll tell you. When if we use directly, to see some object, we use directly 90x, it will give impact. Those radiation will give impact to our eyes directly. That is why what do we do? If we have to see something, we will be starting with the 10 magnifying power. Okay. Now, once we have seen to 10 magnifying power, then we can go to the next one, 15 power. Then we can go to the 45x power. So what is happening slowly and gradually, our eyes are getting used of it, that magnifying power. Clearly, if directly, like if we are directly using the 90, what happened? It gives the radiation which are being harmful. But slowly and gradually, if we are changing the magnifying power, by that time the eyes will get settled down. Am I clear, Geeta, Medha, and Ashrita?
Yes, ma'am.
That is why always we have to use the low magnifying power, then slowly and gradually we have to go to the 90. But generally in our school also, we are using either the 10x, either the 45x. Never ever we are not using the 90x actually. And we do not keep also in the, in that normal kind of a microscope, we do not work for it. So he actually used the magnifying power from the magnification, then focusing screw. With the help of the focusing screw, what they can do, they can adjust the distance between the lens and the specimen. So this is the specimen here, this is the lens here. So what they can do, they can adjust it. The tube-like structure, they can make it up, they can make it down, up, down, so that they can see the object clearly. Then this part where they have kept the specimen, that is called as the stage. So on the stage, like if someone is getting married, they will be standing on the stage, right? Same way, if something we have to see, we have to keep it on the stage so that we can see it. Then oil lamp, basically this is the lamp which is a kind of the light source. Now during that time they do not have such kind of the lenses or mirror and all. Now you have it, right? In the grade 8 you have discussed the light chapter where the mirror, plane mirror, concave, convex, all those you have discussed. But during that time it was not there. So what he did is he just used the lamp actually to lighten that particular kind of the specimen. And then the water also. The reason why he used the water is basically so that the lamp does not evolve so much, or lamp does not illuminate so much of the heat energy. So what happened with the help of the water, the heat energy which is being transferred will get cooled down. It will not create any kind of the hotness on the specimen. Am I clear with this? Any
Yes,
doubt? No doubts. Okay. So let us then one by one see all the parts. So eyepiece, which is also called as ocular lens, the part through which the observer looks. So we are the observer, we are looking through the eyepiece. Magnifying the image formed by the objective lens. So they will be helping the magnifying the image. Then the second one, eyepiece tube. This is called as the eyepiece tube. It holds the eyepiece in position. So because of this tube only, the eyepiece will be holded. And also it maintains the proper distance between the lenses. Then objective lens placed near the specimen. We will be keeping it near the specimen. Then it provides the primary magnification also. So primary magnification means whatever the magnifying power we needed through the objective lens, we can set it. Then the focusing screw, it will be using to adjust the distance between the lens and the specimen. And it also helps in obtaining a clear image. This is very, very important. Like, with the screw, like in your phone, with the help of your finger, what do? You zoom it. You zoom in, you zoom out. So when you zoom it much, the picture get a blur. So what do? You zoom out a little bit. Same way, with the help of the focusing screw, you will be actually rotating. If it is the image is not clear, you will be coming back to the original shape and all. So that is why it is important. Then specimen holder or the stage, it holds the object in place. So with the help of this holder and all, they will be settling down, they will be holding it. Now, light source, which is the oil lamp here, it provides illumination to view the specimen. So basically the light, they will be getting it through the light lamp, light source. Then light is directed towards the specimen. So you can see this light, they will be keeping in such a direction that the specimen get the light. Then water flask filled with water to focus and concentrate on the light, and it acts like the primitive condenser. So kind of a condenser so that the heating up should not take place. Now, why the microscope of the Hooke, Robert Hooke was important? So it is the first instrument used to observe the cell. So it was a very, very first kind of the microscope which was used to identify the cell. That is why it is important. It helps in the discovery of the cell structure, as we know that. And it also helped in mark the beginning of the microscopic study in the biology. So though it was not very much developed, though it was not with so much of the functions and all, but still it was precious, it was important because of this kind of the importance. Now working, how does it work? So light from the oil lamp passes through the water flask. So this light is which is coming, it will be passing through the flask and then it goes to the specimen. So you might have learned about the reflection and refraction, right? What happened in the reflection? So any kind of the ray it enters, it will go back, correct? Yes. Suppose it is the light entering, it will strike and it will go back like this. Same way, the light which comes from the lamp, it will strike the water and water will actually give it to the specimen. Clear? So that is how it was working. Then the objective lens magnifying the specimen and also the eyepiece then further enlarges the image. So through the eyepiece, we can enlarge the image, we can see the image. And if in case still any kind of the improvement is needed, we can use the focusing screw to clarify the image further. So that is how it was working. So some of the fact, which is actually, a kind of negative point for it. So Hooke's microscope produced low magnification images only. So with his the microscope, we cannot see the higher magnifying power like 45x, 90x and all. Only the low magnifying object we can see. Second thing, images were not very clear compared to the modern microscope. So in the modern microscope, we can see the images very, very clear. But in this, we cannot see that. And next thing, though it has so many negative points, but in spite of that, it leads to the scientific breakthrough because it leads to the discovery of the cell. Clear? Medha, Ashrita, and the Geeta.
Yes, ma'am.
Yes, ma'am.
So that is how it was a very, very important, interesting thing it has become. Now, one more topic, let us do it about the living cell. This is very important, children. The name is very important. His working is important. Robert Hooke, he discovered the cell, correct? He discovered the cell which was actually dead in nature. But this scientist, Anthony van Leeuwenhoek, he was the first one who discovered the living cells from the pond water. So, who is Anthony van Leeuwenhoek? Basically, he's a Dutch scientist known as the father of microbiology. Learn this. In your competitive exam, in your Olympiad exam, in your NEET examination, this question comes. Who is the father of the microbiology? So, Anthony van Leeuwenhoek is the father of the microbiology. He designed simple microscope with the powerful lenses. His microscope were more powerful than the Hooke's microscope. Definitely, right? Now it is like earlier something used to be little with less of money, but nowadays it is more of money, right? Now, if I talk about 10 years after, it will be more expensive or it will be less expensive than today.
More expensive.
More expensive, correct? So day by day, year by year, the expensiveness is increasing. Same way, development also from the Hooke point of view from 1665 till this Anthony Leeuwenhoek point of time, what happened? Discovery happened. The things changed. And because of that, he led to the more powerful kind of the microscope. Then how he discovered that? He observed the pond water, rainwater, and the saliva under the microscope. So not only the pond water, he used the other source also, which is the rainwater and saliva. And then what he saw that tiny moving organisms, small moving organisms were there, which he called as the animalcules. Very, very important this is. Animal, animalcules, which means small little animals, which means the microbe. Okay. So this word they generally ask, who discovered the animalcules? It was discovered by the Anthony van Leeuwenhoek from the pond water. These were actually living cells, and they were first observation of the living cell, which was done by the Anthony van Leeuwenhoek. So that is how it was led to the discovery. Clear, Geeta, Ashrita, and Medha?
Yes, ma'am.
Yes, ma'am.
Okay, now coming to the next one, importance of his discovery. So, with this discovery, with his information, he proved that the cells can be living and active. Now, if something was moving, definitely we will be saying it as living only. Now, if something is living, it can move. Now, some organisms can move with more activeness, some can move with little less activeness. So, based on that, he proved that the cells are living and they can be in active form as well. It is not only they can be at one place, they can move. And he opened the field of microbiology. So when he discovered the living cell, what happened? Animalcules are basically little animals. Little animal means the microbe. So with his discovery, the doors for the small microorganism or for the doors for the microbiology study has been extended. So he opened the field of microbiology. After him, many other scientists came who actually worked on the different types of the small animals, the microbes, virus, fungus, bacteria, all those things. It also helped the scientist understanding microscopic life form. So microscopic life form means everybody has different. The virus has a different form, right? They can survive in the hot climate or cold climate, or what do they need? They need the host. So like this, the bacteria has different climate. The algae can live only the water. So because of his discovery, he was actually able to identify what kind of the area, what kind of the surrounding do they need. Then it led to the further discoveries about bacteria and protozoa also. So when he started studying about the microorganisms, ultimately he got to know about the bacteria also. That something called bacteria also exists in our nature. He also got to know about the protozoa like amoeba and all. They also survive actually. So likewise, all these things were connected. Robert Hooke started from the dead cells, but later on, because of his developed microscope, it led to the discovery of the other forms of it also. Clear? So this is how the discovery of the living cell was done. Rishit, you have joined the class late. Why, Rishit?
I'm not in my home. I'm like out of town, so I have some issues with network.
Okay, no problem. Yeah, any doubt, Ashrita, Geeta, Medha, and Rishit now?
No, ma'am.
No? So very important. Learn the things. Don't mug up anything. Just understand. Robert Hooke, he discovered the dead cell. The Leeuwenhoek, he discovered the living cell. Dead cells were discovered from the cork part, from the bark part of the tree. The living cells were discovered from the pond water. The living cells were named as the animalcules, which means the little animals. And who is the father of the microbiology?
Anthony Van Leeuwen.
Antony van Leeuwenhoek. Very good, very good. Then about this cell theory. After this, now what happened? Other theories were also there, other points were there, other discoveries were also there. In the other discovery, there's another part which is called as the nucleus. Do you know what is the nucleus?
The brain of the cell.
The brain of the cell. Very good. Okay. Any other point? Medha, Rishit, and Ashrita? Any other point regarding the nucleus? Any idea about that? Yes or no? Medha, you have any idea?
Ma'am, I couldn't
No.
hear you about.
Ma'am, I know what is it, but I don't know how to explain.
Now am I audible, Ashrita?
Yes, ma'am.
Okay. I was asking, do you know what do you mean by the nucleus?
Nucleus. Yeah, I don't have an idea about it because I had it last year. Nucleus is the smallest object, and it is the main part.
Okay, fine, but not the smallest one. We cannot say it as the smallest one. It is round spherical structure, but yes, main part of it which controls everything. Like we have the brain, right? The brain will be controlling. Brain will be deciding when I have to speak loud, when I have to be slow, when I have to shout, when I have to talk with you, when I should not talk with you. All those things my brain is controlling, right? Same way in our body, we have the cells, and inside the cells, there are the nucleus who controls all the activities of the cells. So many parts are there inside the cells, and everything in that cell will be controlled by whom? By the nucleus. Now, that nucleus was discovered by the scientist known as the Robert Brown. Okay, Robert Brown is a scientist who discovered the nucleus, who mentioned that the whole entire structure of the nucleus. So in the upcoming classes, we will be discussing the entire structure of the nucleus as well. But for time being, you understand that it was discovered by whom? By the Robert Brown. So he explained each and every part of the nucleus, the ball, the cover, inside what kind of structure is there, what all things are present inside, everything he has explained in a detailed manner. Okay, so Robert Hooke was the person who discovered the nucleus. Apart from that, there is another scientist known as the Purkinje. 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 So Purkinje, what he did, he gave the term which is called as protoplasm. Protoplasm, I think you have never ever heard. Have you heard that protoplasm word?
No.
Geeta? No. Rishit is saying no. Ashrita? Have you heard the protoplasm? No, right? Okay, I'll tell you what it is. When we talk about the protoplasm, it means the cytoplasm and the nucleus together. Okay, two components which are the cytoplasm and nucleus, when they join together, we call it as a protoplasm. So protoplasm is the living component of the cell where the cytoplasm and nucleus are present together. Now what is cytoplasm? Any idea?
The jelly-like structure around the nucleus.
Correct. So around the nucleus, or we can say inside the cell. Okay.
Inside
So
the
inside the cell, we have the jelly-like structure, which is called as cytoplasm. This cytoplasm, what it does, it actually hold the parts of the cell at specific place. Okay. Now, for example, if this imagine if we do not have any kind of the cytoplasm, okay. Now I'm bending like this. What happened? All of my content will go that side. Now I'm bending like this. Everything will go now this side. Correct? Something should be there to hold it. Who will be doing that? That will be done by the cytoplasm. So cytoplasm is jelly-like structure which help to hold the other parts of the cell at a specific place. Clear? So cytoplasm and nucleus together, we call them as a protoplasm. Any doubt, children, in this now?
No, ma'am.
Rishit, any doubt?
I have a doubt with the previous
Tell
one.
me.
Discovered nucleus.
Huh?
Like the person who discovered nucleus. In our school, they said Rutherford discovered nucleus.
Okay. Rishit, that nucleus is different. That is about the physics.
Okay.
Okay, that is physics. Here it is the bio.
Okay, I mean, like here it's cellular nucleus and there it's atomic nucleus.
Correct.
Okay.
Correct. Okay.
Okay.
Very smart, Rishit, but so Rutherford has given the different concept related to the nucleus, wherein this nucleus, when I'm talking about, it's related to the cell of our body.
Okay.
Okay. So, okay. Then let me share the screen. Now, what do you need to do? You have to. Today is 25.
Yes, ma'am.
Okay, and Wednesday. And also, it is the cell L2. Now, I'll just give me a minute. Let me paste one image here. Just a minute, okay? I'll just stop sharing the screen. So very easily you can write it. Okay, children, so you can start writing this part now.
Ma'am, I actually
Tell me, Ashrita.
nothing, ma'am. For a second, I couldn't see anything there. That's why.
Okay, fine. So you can start writing this part. In this, what you need to write, I'll tell you. You have to write about the parts of the microscope and their function. And the heading, what you need to write is the Robert Hooke's microscope. So
Mute.
write the heading Robert Hooke's microscope, and there write the about the parts 1, 2, 3, 4, 5, 6, 7.
See a screen.
It's not visible?
No,
Mute
ma'am. No,
camera.
ma'am.
Okay, let me share it again. And bio children, it is very, very scoring and very easy subject. If you like it, that is also the point. If you like it, it is very easy. And I know you all like it, right? So these are the 7 points which you need to write, children. Start writing now.
Ma'am, can you zoom it a little bit? Enough. No?
Ma'am,
Yeah, tell me.
ma'am, what do we have to do?
We need to write these parts. Only the parts and the function you need to write, okay?
Okay, ma'am.
Rest, I will be sending this entire poster in the group. But this parts and the functions are important. That is why you need to write them. Other things you can go through and you can just read them, okay? And your homework will be to complete
Okay,
this.
ma'am.
Your homework will be, children, to complete these parts and the functions. And also, I want you to tell me tomorrow in the class about the compound microscope and the light microscope. Are they same? Are they different? Okay. So difference between the compound and the light microscope that you need to find out, and you need to tell me tomorrow in the class. So I'll share this on the WhatsApp also. Just a minute. So Geeta, Medha, Ashita, Rishit, you wanted to ask anything? Any doubt do you have?
No, ma'am.
And how the maths class is going on? How was the physics class?
Well, ma'am.
Any kind of the concern, any kind of the suggestion?
No.
And children, make sure that you complete the homework and send it then and there for the correction part. And I think Friday and Saturday you will have your chemistry class also, okay? And social class also you might have on Friday or Saturday. So slowly and gradually everything will be, fine within this week time. And this Sunday you will have the test of the bio, wherein whatever we have discussed, we will be discussing till tomorrow. That will be there in your portion. So it can be anything. It can be any kind of. Sorry, Geeta, come again.
Is the test objective or subjective?
I think subjective. What I've given, I can't
Just
remember. I think subjective I might have given.
okay, ma'am.
Subjective test. It is subjective.
Okay, ma'am.
Okay. So wherein I can, in the subjective test I can give you the assertion reason also. I can give you might be the MCQs also. So for me subjective means wherein few MCQs, few assertion reason, very short. It can be in that way also, or it can be the entirely name the following.
Yes, ma'am.
Okay. Fine, children. Then tomorrow I think we have the class at the same timing, and we'll connect soon then tomorrow. And tomorrow we will be discussing about the cell theory. Cell theory, again, a very, very important topic, an interesting topic. After the discovery, the different scientists came. They gave their own points. Like, it happens in the family also. Mother will have the different thought processing, father will have different thought processing, you will have the different thought processing, and everybody will have come with their thoughts and all. And ultimately, it leads either to the solution or it leads to the confusion. Correct? So when you go for the shopping, you get confused. You will be saying, "No, Mama, I need this one." Your mother will be saying, "No, beta, you get this one." Your father will be saying, "Do anything. I don't care. You take anything." Right? So that confusion that happens in the cell also. Different scientists were there. They have given their different theories, and based on their theories, different outcomes were there. So that we will be discussing in the tomorrow's class. Okay?
Okay, ma'am.
Okay, children. Then thank you.
Hey, ma'am, thank you, ma'am.
Thank you.
Thank you, ma'am.
And complete the homework, write it, and send it. Okay? By
The
today.
Okay, ma'am.
difference one, should I write it down?
Sorry, come again, Geeta?
The difference homework, should I write it down?
Yes.
Okay, ma'am.
Okay, thank you.
Bye, ma'am. Thank you,
Bye
ma'am.
bye.